Electron transport system. Let's understand this topic today. As the name suggests, it's a system of various complexes which carry out the transport of electron. This is a part of oxidative phosphorylation because the phosphorylation of ADP takes place with the help of the oxygen. Here, the oxygen is the final electron acceptor along with the hydrogen molecule and it forms water molecule. During the transport of electron, H plus ion moves across the membrane from one place to another place, and meanwhile the high energy compound are converted into the ATP. The high energy molecule is nothing but NADH and FADH2. Now let's understand what are this NADH and FADH2. We have already studied the process of glycolysis. From the glucose, two molecules of pyruvate are produced during the process of glycolysis. Meanwhile, NADH are generated. So this NADH are high energy molecule. Similarly, from pyruvate to acetyl coenzyme, again NADH are generated. When the acetyl coenzyme enters the TCA cycle or citric acid cycle, again during the intermediate molecule formation in between NADH and some molecules of FADH2 are generated. These NADH and FADH2, these are the reduced form. They act as electron carrier. How they act as electron carrier? These NADH reduced form will oxidize to NAD plus H plus will donate electrons. This reaction takes place inside the mitochondrial matrix. So let's see the mitochondrial matrix first. This is the structure of mitochondria. We know that mitochondria is a double membrane structure. This one, brown color, is the outer mitochondrial membrane. This sky blue color is the inner mitochondrial membrane, and the pink dots inside the inner mitochondrial membrane, this region is known as mitochondrial matrix. So, the NADH and FADH2 are present inside the mitochondrial matrix. And these NADH and FADH2 must be reduced, they are reduced form, so they must be oxidized and gives electrons. So the electron transport system, most of the components of electron transport system are present in the inner mitochondrial membrane. The components are 5 complexes: complex 1, complex 2, complex 3, complex 4 and complex 5. Out of which only complex 2 is present in the mitochondrial matrix. Rest all 4 complex 1, 3, 4 and 5 are present in the inner mitochondrial matrix. Now, the NADH that is produced during the glycolysis or the TCA cycle, which is present in the intermitochondrial membrane, when it gets oxidized, it forms NAD plus H plus and 2 electrons are given. These 2 electrons are accepted by flavin mononucleotide. Here I have drawn a female representing flavin mononucleotide or FMN. So this FMN accepts these 2 electrons and it passes this 2 electron to FES. This FES is iron sulfur protein. As we know that iron is a strong iron, we represent iron as a strong compound. So I have shown this, So I have shown this hammered like structure, strong structure. So this electron are accepted by this FES protein. This FES protein transfers the 2 electrons to the ubiquinone. Now this ubiquinone is again reduced to ubiquinol. Ubiquinone is oxidized form, ubiquinol is reduced form. So when this ubiquinol again gives 2H plus, passes 2H plus to the intermembrane space, and these 2 electrons are passed on to complex 3. Here in complex 3, it is given to cytochrome B, then to again iron sulfur protein. From iron sulfur protein, it is again given to cytochrome C1. From this cytochrome C1, the electrons are passed to cytochrome C. This cytochrome C at a time pass 1 electron to complex 4, that is cytochrome A, and ultimately to cytochrome A3. So here we can see how the electrons are passed from NADH. From NADH, 2 electrons to FMN, FMN to FES, FES to ubiquinone. Ubiquinone is reduced to ubiquinol. From this ubiquinol to complex 3 of cytochrome B, cytochrome B to iron sulfur protein. From there to cytochrome C1, and from cytochrome C1 to cytochrome C, I have drawn the C like structure, so you can easily memorize. The cytochrome C to again complex 4, cytochrome A and cytochrome A3. Now these electrons are finally accepted by atomic oxygen, that is 1/2 O2 plus electron plus 2H plus, and all together it will form water molecule. Now let's understand about the movement of the H plus ions. During the movement of 2 electrons through FMN and FES and ubiquinone, the 4 hydrogen ions passes through the inner membrane and moves to the intermembrane space. Here once again let's see, this is the inner membrane, this is the outer membrane. So this much region is nothing but the intermembrane space. So 4H plus moves from matrix through inner membrane to the intermembrane space. Again, when the ubiquinone is reduced to ubiquinol and again ubiquinol is oxidized to ubiquinone, 2H plus ion again liberated to the intermembrane space. Similarly, when the electron pass through complex 3, again 2 hydrogen ions are also passed from matrix to the intermembrane space. Finally, passing of electron through complex 4, again during that 2H plus ion passes across the inner membrane and moves to the intermembrane space. So we can see here that if the NADH is the electron donor, 4H plus, 2H plus, more 2H plus, total 8H plus, and this 2H plus. So total 10H plus hydrogen are here in the intermembrane space. Now there is a role of complex 5. The name of the complex 5 is ATP synthetase. We know that ATP synthetase is made up of 2 components, F0 and F1 particle. Here F0 is embedded in the inner membrane, whereas F1 is protruding towards the matrix. Here you can see. So this is the matrix. So F1 is towards this side. Now 10H plus are to the intermembrane space, from where 4 hydrogen ions, when they pass through the channels of ATP synthetase and again come back to the matrix, ADP is phosphorylated and forms ATP. So here ATP molecule is generated from the transport of 4H plus ion ATP molecule is generated. So, 4, 4 and 2. Total 10 H plus are there. So, total 2.5 equivalent to 3 ATP molecules are generated, because after every 4 transport of every 4H plus ion, 1 ATP is generated. So, again 4, 1 more ATP and 2.5 or half ATP. So, 2.5 ATP after the transport of 10H plus from the intermembrane to the matrix, total 2.5 equivalent to 3 ATP are generated. Hope you understand. Now let's talk about complex 2. We know that during the TCA cycle, acetyl coenzyme A enters to the TCA cycle and various intermediates are produced, out of which one is succinate, from which fumarate is produced. Now the enzyme succinate dehydrogenase is present in the matrix. So when the succinate is converted to fumarate, 2H plus ion are produced. These 2H plus ion again passes across the inner membrane and again they move to the intermembrane space. Here there is no role of complex 1. So the role of complex 1 is eliminated. So 4H plus, when the FADH2 is the electron donor, the 4H plus, this 4H plus are not there in the intermembrane space. So when the FADH2 is the electron donor, out of 10 only 6 hydrogen are present, 6 hydrogen ions are present, protons are present in the intermembrane space. So when 6H plus moves across the membrane towards the matrix, again 1.5 ATP is generated, which is equivalent to 2 ATP approximately. So when NADH is the electron donor, 3 ATP is generated, and when FADH2 is the electron donor, 2 ATP is generated. This way, the electrons are transported through various complexes. Complex 1 name is the NADH dehydrogenase because it removes the hydrogen from NAD. Complex 3 known as cytochrome reductase, it reduces the cytochrome. Complex 4 cytochrome oxidase, it carries out the oxidation, and complex 5 is ATP synthetase. Here the final electron acceptor is oxygen molecule. Oxygen atom. So oxygen, the role of oxygen in aerobic respiration is during this oxidative phosphorylation. So here we understood that from the high energy molecule FAD and NADH, they carry out, they transfer the hydrogen ion. Meanwhile, they also transfer the electrons from one complex to another, from one electron carrier to another electron carrier. They carry out the oxidation reduction reaction, and ultimately the electrons are accepted by O2 and water molecule is generated, and whatever the hydrogen moves from matrix to the intermembrane space. Meanwhile, there is a creation of proton gradient, due to which again these protons should be pumped back via ATP synthetase. Meanwhile, again by the transport of every 4H plus, 1 ATP molecule is generated. Here we have to notice that the complexes, they work or they transport the electron on the basis of the redox potential. The electrons move from low potential to high potential. So Complex 1 is obviously has low potential as compared to the complex 3, and complex 3 has low potential than complex 5. Sometimes we encounter the term equivalent, redox equivalent, that means again the electrons. Hope you understand this reaction. If you want to memorize this components of electron transport system, I will give you the tricks. The trick is a story. There is an area known as NAD area in which the females are very strong like iron. So they, due to their strong nature, they became queens. That is ubiquinone. Another name of ubiquinone is coenzyme Q. Here I use the word ubiquinone for the queens. Now these queens, they created a system, cytochrome. They created a system that the females have to be strong like iron to see the system again, that is the cytochrome. So when they will become strong like iron, the entire world will be ready to make them A1. This is cytochrome A and A3. So they will be A1. And the strength is also interrelated to the word ATP or energy. So hope you like this video, and let me know in the comment box how you liked it, and if you want to understand some other concept, please do comment in the comment box. Thank you.